Okay, so welcome to the second mini lecture in this series of extensions of Mendelian genetics. Let's start from the first slideshow. So this lecture covers then the 4.4 section in your text. Basically, it's looking at other different versions of inheritance that you can see when you're looking at a single gene. So basically what we're looking at here are one gene and how the two alleles can sort of fight it out to give you different phenotypes. The first we're going to look at is called incomplete dominance. That is when the heterozygote the heterozygote genotype exhibits a phenotype that's kind of halfway or intermediate between the recessive and the dominant phenotypes. This is actually an example of haploinsufficiency that I talked about in the last lecture. So an example of this, and it's easiest to figure it out when you look at an example, is flower color in a plant called the 4 o'clock. So again, we're looking at one gene here. And if you look at, if you look at the phenotypes of this plant, you see that you can have a white flower, a red or purple flower, and then one that's kind of halfway in between two. This is an example, this pink flower, of incomplete dominance. So let's have a look at how that works genetically. We're talking about one gene. We're going to call that gene C for color. And there's two different alleles. This time, instead of the alleles being big C and little c, we're going to denote them as C superscript R, which is the wild type allele for a red flower color, and C superscript W, which is the allele for white flower color. Now, this case, in other words, incomplete dominance, is a good example of where 50% of the protein function does not rescue 100% of the phenotype. So previously, when we looked at this, we saw this example of classical Mendelian genetics, where 50% of the functional protein, big P, little p, rescued 100% of the phenotype. That is not what occurs in incomplete dominance. So how does that work from a genetic perspective? So we'll take two true breeding 4 o'clock plants. One of them is red, one of them is white. True breeding. So they have a homozygous situation. The red flowers are homozygous for the dominant gene, which is 100% of the protein. CR, red. In this case, you have a homozygous recessive situation, CW. This contains 0% of the protein, in other words, a pigment in this case, a red pigment. Therefore, the flower is white. Now, if you cross these two guys together, the gametes are obviously just CR for this one and CW for this one. And when these combine, give you a CR-CW plant in the F1 generation, it's pink. Because in this case now, we have 50% of the protein. 50% of the red protein gives us a not quite as red plant, which you see phenotypically as a pink plant. That is incomplete dominance. Now, incomplete dominance gives you a slightly confusing result when you look at the F1 self-cross. So remember that this is what Mendel did a lot, right? He took homozygous, true breeding plants, crossed them to create a heterozygote, cross that to itself, so across the F1 generation to itself, which gives you the F2 generation, which had a classic 3-to-1 ratio. Well, you don't get that in the term in incomplete dominance because you get the same Punnett square in terms of a genotype, right? So we still get a ratio of 4 things, but in this case, the phenotypic ratio is different. So the genotypic ratio is a quarter of the progeny will be homozygous red or homozygous CR. A quarter of the progeny 1 in 4 will be CW, homozygous recessive, and 2 out of the 4 will give you a heterozygote, right? Remember, in classic Mendelian genetics, all 3 of these would look the same, and this one would look different, giving us the 3-to-1 ratio. What you see in this case is the homozygote dominant is red, the homozygote recessive is white, and the heterozygotes, because this gene is showing incomplete dominance, are pink, which gives us a 1 to 2 to 1 ratio. Now, importantly, you add these up, right, and you still get a ratio of 4. That tells you that one gene is involved, and that gene has 2 alleles. Just that this time we're looking at incomplete dominance, not classic Mendelian complete dominant situation. Another interesting genetic phenomenon that occurs is something called overdominance. Now, this is a case in which the heterozygote is actually more vigorous in some way than both of the corresponding homozygotes. So, for some reason, the heterozygote gives you a more, makes you as an organism more efficient or better, in some way more vigorous than both a homozygous dominant or a homozygous recessive situation. Because of that, this is often called heterozygote advantage. And an example is something you already know a little bit about, which is sickle cell disease. So remember that sickle cell disease, right? You have a person that doesn't, a normal red blood cell looks like this, and a sickled red blood cell like this. And if you recall, this disease is caused by a single mutation that acts in a typical, acts as a typical Mendelian trait in terms of whether or not you get sickle cell issue, right? So it's a single gene. In this case, we have hemoglobin A, hemoglobin B. This is a heterozygote for sickle cell versus normal, S being sickle cell. And you're doing a cell cross with another heterozygote, so you get a classic Mendelian ratio of, here we have the homozygote dominant, homozygote recessive, and the heterozygotes. Interestingly, in this case, you know that these 3, in terms of sickle cell disease, are going to be normal. These are not going to have sickled red blood cells. This last one will, because it's a classic Mendelian ratio. But interestingly, if you are a heterozygote for this gene, like these 2 people are, then you actually exhibit a resistance to malaria. So that's a bonus that you get for being a heterozygote. That selective advantage explains why a lot of African Americans, or well, not a lot of African Americans, that's the wrong way to put it, but sickle cell disease is more predominant in the African American community than it is in the Caucasian community. And that is because there's a lot of malaria in Africa. So in Africa, there's a selective advantage for these heterozygotes. And that is an example of overdominance, where the heterozygotes have some kind of selective advantage over the homozygote dominant, or indeed the homozygote recessive. Finally, we're going to talk about codominance, which is how blood typing works. And for this, you need to know a little bit of sort of what I was talking about at the start of the last lecture, which is just that. It's true that many genes have many, in other words, 3 or more alleles at the population level. So what does that mean? What that means is any individual, like me or you, can have 2 copies of each gene. Hi, this is my daughter, Elsie. Elsie, I'm recording a lecture right now, so could you give me a few minutes, please?
I need to tell you something.
Okay, what do you need to tell me? Oh, good point. We will do that in a minute. Okay, you got to leave me alone right now, though. Thank you.
What did you tell?
So, sorry about that. it is true that me or you can only have 2 copies of each gene, which is, one from mom, one from dad. But in a general population, if you look at our class in general, for any given gene, there's probably a lot more than just 2 possibilities, but only 2 possibilities can exist within a single individual, at least in a diploid organism. And depending on which 2 alleles you have, they can interact with each other in weird and wonderful ways. One of the ways is codominance. We've already talked about a couple of others. And if you look at the A, B, and O blood grouping situation, that is an example of multiple alleles that can be codominant. I'll explain what that means. Yeah. So, as I said, in many cases, there's more than just 2 alleles in a general population. In fact, there are 3 alleles that are floating around in the population that determine your blood type, whether you're type A, type B, or type O. And those 3 alleles are called capital IA, capital IB, and lowercase I. Remember, any individual, so we're talking about one gene, the gene is I, and we have 3 possible alleles, of which any individual can only have 2, because it's the same gene. So, what does codominant mean? The fact that the I is lowercase should be a good indication to you that that is a recessive allele, right? That's the convention when you look at genes. So this allele is recessive. These guys, IA and IB, are both capital I. And that means that they are both dominant over this little I. So both IA and IB are dominant to the I allele. What does that mean? If you have either of these and an I allele, then whichever one you have will be dominant over the I allele. So what if you have both of these? They are co-dominant when both present in the same person, which means the protein from each allele is expressed equally, and you see both phenotypes of each allele, equal numbers or in equal quantity in the individual. So let me explain what that means, because it basically is the reason why there are 4 possible blood groups. Blood group type A, type B, AB, or type O. So I said that you can only ever have 2 alleles of one gene, right? And that's generally true. So if you are either homozygous IA or a single IA allele and a recessive allele, your phenotype, blood type in this case, is type A. To be a type B, you have to carry 2 dominant B alleles or a dominant B allele and a recessive allele, and then you're type B. If you carry both type A and type B alleles, they are co-dominant. Both are expressed equally. You see both phenotypes in the organism. Your blood type is AB. If you have 2 recessive alleles, then you're neither type A or type B, you're type, or even AB, you're type O. And that's how you get 4 different phenotypes or blood types from depending on what combination of these blood typing alleles you get in your body. So that's all I want to say for this part of the class. Look at the book for any expansions on this stuff, or drop me a line. Okay, thank you for listening, and on to the next one.